Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So, we were learning about cancer biology, in which we were discussing about hallmarks of cancer. So, we have already covered three important hallmarks of cancer: how the tumor cells they are able to sustain a proliferative signals, how they evade growth suppression, and how they are able to evade apoptosis. In today's lecture, we are going to learn about replicative immortality, how the cells they are able to sustain they do not enter into the replicative senescence stage, and why, how do they evade this stage. Okay, so today we are going to learn about a very important hallmark of cancer, how that is exhibited, what are the mechanism to exhibit that hallmark. So the we are going to learn about molecular mechanisms of replicative immortality, which is exhibited by tumor cells. So for that matter, let's learn about what is replicative immortality. Okay. So I hope you are aware about end replication problem. So whenever cells undergo cell division, the 3' end of the DNA that is not replicated properly in the lagging strand, and this problem is resolved using the telomerase enzyme. But in 90% of the cases in the somatic cells, they lack telomerase enzyme. So the ends of the DNA they are not they are not replicated properly, and this results into telomere shortening. So eventually telomere length decreases, and at that point of time the cells enter into a stage where it stops dividing, and that stage is known as senescence stage. So this stage is very important because it is a very reliable mechanism to prevent cancer cells from proliferating, because cancer cells are uncontrollably growing cells. So once you activate the senescence stage, now the cell cannot divide. So this prevents the tumor formation. So replicative capacity and telomere maintenance in cancer cell is quite different from what we see in normal cells. So this is a very defining hallmark of cancer, which allows malignant cell to sustain an uncontrolled proliferation. So this is achieved, right, like the replicative immortality. This is achieved through telomere maintenance mechanism. So in this lecture, we will also learn about telomere maintenance mechanisms. So for that, let's first understand about what is telomeres. Okay. Telomeres. So let's learn about the telomere biology. So what do telomeres do? So telomeres, they are specialized nucleoprotein structures that are located at the ends of the chromosomes. So the functions of telomeres are as follows. So they protect the chromosomal termini from degradation, fusion, and aberrant recombination. So due to, they are the nucleoprotein structure. So that protein which fuses with the nucleic matter at the end, this prevents into the degradation, because sometimes that can be considered as DNA double stranded breaks. So this prevents telomeres, they prevent degradation, the chromosomal degradation by enzymes. Okay, and the chromosomal fusion, as well as the recombination, the aberrant recombination. They protect them from all those things. Okay, so chromosomal ends can fuse. So that fusion is also avoided with the helps of telomeres. So telomeres, they also, maintain, they preserve genomic stability, which could be disrupted if those ends, the ends of the chromosomes, they are recognized as DNA double-stranded breaks, because DNA, it ends somewhere, right? So wherever DNA ends, this can be considered as DNA double-stranded break. So telomeres prevent the recognition of those ends as double-stranded breaks by the enzymes. So this is prevented, and this helps in preserving genomic stability. Again, telomere, they serve as the mitotic clock. So as the telomere length keeps shortening, it shortens the cellular lifespan. So telomeres, they also regulate cellular lifespan. length of telomeres directly convey about the lifespan of the cell. So once the telomere shorten enough, this will shorten the cellular life, because cell is now going to enter into the senescence stage. So well, most somatic cell, they undergo telomere shortening. The cancerous cell, they have a mechanism to, to prevent the telomere shortening, which eventually leads to replicative senescence. So how do they do that? So cancer cell, they bypass this limitation by reactivating telomere maintenance mechanisms. So there is something which is known as telomere maintenance mechanism, TMM. I'll be writing it as TMM. So those helps into, reactivating this helps in maintaining the telomere length, so telomere would not be short. Or cancer cells, they re-express the telomerase, which is usually, silenced in the most of the cells. So they can reactivate or express the telomerase enzyme. This is an enzyme which helps into, maintaining the telomere length. So by doing the TERT activation, the TERT, telomerase reverse transcriptase, this is a catalytic subunit of telomerase enzyme. We'll learn about this as well. So or they you can go for ALT pathway. This is the alternative lengthening of telomeres pathway, which do not use telomerase for for maintaining the telomeres length. So this three pathways, these three mechanisms can be adapted so that the telomere length would not be shortened, and cancer cells, they can exhibit the replicative immortality. So let's first learn about telomeres biology. So we learned about what do telomeres do. Let's first learn about the composition of telomeres. So telomeres, what are they composed of? So they are composed of the tandem repeats. Okay, so this is a repetitive sequence which is found in DNA, which which is expanded few kb. Like the size of these repeats is like few kb. So in the sequence of this tandem repeats is different in different individual. In humans, it is TTA GGG. So this is the sequence of tandem repeats that are found at the telomeres in humans. Okay. Then telomeres, they also have a single-stranded G-rich overhang. So they have a single-stranded G-rich overhang. So this G-rich overhang, this forms a protective T-loop structure. Okay, so this protects it from fusion, from degradation, and all. So this forms a protective T-loop structure. Okay, the single-stranded G overhang. And then besides those sequences, telomeres have some binding proteins which form a sheltering complex. Okay, that is called as sheltering complex. Okay, so these proteins are quite important because they are the ones which are going to protect the telomeres, the ends of the chromosomes. So these are of different, they belong to the different families like TRF1 and TRF2. These are telomeric repeat binding factors, TRF1 and TRF2. They're involved in telomere length regulation. So they do the telomere length regulation. Then there is RAP1, repressor activator protein 1, which prevents telomeric fusion. So there is RAP1. This prevents telomeric fusion. So that telomeric ends, they will not fuse together. Then there is POT1, protection of telomeres 1. This binds to the G-stranded, the G overlap. So this binds to the T loop. The T loop, which is formed by the G overhang, so the single-stranded G overhang. And there is again TPP1 and TIN2, TIN2. Both of them, they serve as adapters This adapters, they stabilize the sheltering complex. So these are the proteins which are found at the telomeric length. So telomeres, they are actually the nucleoprotein complexes, right? They are nucleoprotein structures which are found at the ends of the chromosome. So that ends have tandem repeats, a single stranded G overhang which can form a T loop, and a sheltering complex that is composed of the different types of proteins. So those proteins are listed down here. So this is what telomeres is composed of. This this helps. This will be helpful because the this tells us how they are able to protect the ends. So we already know that telomeres, the telomerase enzyme that solves the end replication problem. So end replication problem is where the DNA polymerase, it cannot fully replicate the 3' end of the chromosome, because in the lagging strand, when the RNA primer is removed, at the end of the whole replication, there will always be a sequence remained at the 3' end, which will not be replicated, and that is solved by telomerase, because the telomerase enzyme that is going to do the reverse transcription of the template DNA, and it will increase the length of template DNA, so that then the FEN1 enzyme will cut in such a way that the junk DNA will be cut and not the important DNA. So we'll learn about end replication problem and how the telomerase enzyme solves it in some other lecture. So if you don't know, please visit that concept. Okay. So let's learn about how the telomere shortening and replicative senescence, that is related to the cancer suppression. How do they help in stopping or inhibiting cancerous cell to proliferate? So we know that with each cell division, telomeric DNA will be lost due to those limitations of lagging strand synthesis. Then once the critical threshold of that telomere length is reached, the telomeres, they will lose their protective function and they will expose the chromosome ends. Okay, so now as the protective sheath has lost, the chromosome ends, they will be recognized as DNA double stranded breaks. And whenever DNA double stranded breaks are they are recognized, it will activate the DNA damage repair pathway. So it will activate the P53 tumor suppressor pathway and PRB tumor suppressor pathway. I have already explained those pathways in previous lectures. Please go and visit that. So those tumor suppressor pathway will be activated. So the DNA damage response activation will be done due to the telomere length shortening. So let's list down the steps. First, telomere length shortens, and it reaches to certain threshold level, certain critical level. Now, when it reaches to its critical length, the DNA damage response will be activated. Why? Because the telomeric ends will be recognized as the double-stranded DNA breaks. Okay. So once the DNA damage response is activated, the P53 and PRB pathways activated, it is going to do the arrest. It will arrest the cell into senescence stage. So it will cross the senescence checkpoints. So it will do the P21. I've explained that in the P53 pathway, that the P21 mediated cell cycle arrest will be seen. So now cell cycle will be arrest in a stage where it cannot replicate anymore. So DNA replication will be inhibited forever. Okay, so cell cycle arrest will be seen if the P53 and PRB pathways are activated. But let's say due to some reasons, those senescence checkpoints, they are bypassed. So let's say that P53 and PRB pathway, they gets inactivated due to this could happen due to multiple reasons. So if those pathways doesn't get activated, what happens is that telomere length will keep shortening. Why? Because now cell is dividing on and on. The cell division is going on. So we already know that cell divides till the Hayflick limit, right? So like only cell divisions will be only till to 40 to 60 divisions, and then cell will it will enter into cell cycle arrest phase. I mean. So once the 40 to 60 divisions are done, it will the cell cycle will be arrested. But if, let's say due to some certain reason cell cycle arrest does not happen, P21 doesn't get activated, or the P53 and PRB pathway doesn't get activated. So when this happens, cell will keep dividing. This means the cell division is going on. So when cell will keep dividing, the further loss of telomere will result into the end-to-end chromosomal fusion. So now telomere will lose itself completely, and this will result into the chromosomal fusions that can lead to catastrophic mitotic failure. And this mitotic failure will lead to apoptosis that we have learned in previous lecture as well. So if this pathway doesn't get activated, it could lead to apoptosis. And once cell dies, this ensures that the genomic instability, whatever was accumulated over in that cell, will not be, will not be, it will not be, like it will not be transferred to the future generations. So it will not be accumulated in the individual's body. So this ensures that the cancer initiation does not take place, and this will ensure that the normal cell will not be converted into tumor cell. But cancer cells, which have accumulated certain accumulation, let's see how the cells accumulated certain mutations, which helps them to convert from normal cell to tumor cell. So when a normal cell shows the some mutation into the telomerase gene, that will lead to the conversion of a normal cell to a tumor cell. So let's see what happens in cancerous cells. So in cancer cells, they show the telomerase activation. So telomerase, this is an enzyme which is going to solve the end replication problem. So this is the enzyme that is going to keep the telomere length constant, like will prevent the telomere shortening. So this enzyme is silenced in most of the somatic cells after early development. But in the 90% of human cancer, tumor the telomerase re-expression. Telomerase has a subunit known as TERT. This subunit re-expression is seen in most of the cancer cells. Like 90% of human cancers show the re-expression of this TERT catalytic subunit which results into the telomere elongation, and this is how the cancer cell they show the replicative immortality. So let's first learn about telomerase enzyme. So this telomerase complex has different subunits. The first one is TERT, which is known as telomerase reverse transcriptase. So this is the catalytic subunit. And then it has an RNA component as well, which is written as TERC. So this is telomerase RNA component. So this RNA component, this provides an RNA template for the telomere elongation. So RNA template will be provided, and then DNA polymerase will elongate that part and will create a junk DNA, which will be cut by the FEN1 enzyme. And this is how we maintain the original DNA length. Then there are certain proteins, the accessory proteins that are going to stabilize the telomerase complex, such as discarin, okay, NOP10, GAR1, and NHP2. So these are the accessory proteins that are going to stabilize the telomerase complex. So telomerase is a complex, so this enzyme is a complex of a reverse transcriptase, an RNA component, as well as certain accessory proteins. Okay. So this is the composition of telomerase enzyme. So let's now learn about the mechanisms by which this TERT reactivation or re-expression is done in cancer cells. This could be done by multiple mechanisms. The very first mechanism is the promoter mutations, mutating the promoter of the gene which is going to express TERT. Okay, so what happens is that there are certain promoter mutations, like the cysteine at the 228 position is converted into threonine, or cysteine at 250 position is converted into threonine. So those two mutations, the mutations like those within the core promoter region of TERT, this creates a de novo binding site for certain transcription factors. The transcription factors which belongs to the ETS family. So ETS family transcription factors. So now what happens is that this particular mutation, this will create a de novo binding site on the DNA for those transcription factors, those ETS transcription factor. The specific one, the very important one is the GA binding protein, which we write as GABP. This is GA binding protein. This is a very particular transcription factor which leads to the TERT activation. So let's say how this GABP leads to the TART transcription. So this GABP has two subunits, alpha and beta. The alpha is DNA binding subunit. Okay, and beta is the transactivation subunit. So alpha DNA binding subunit and beta is transactivation subunit. So in this beta, the beta subunit also has different, it also has different parts. In that the GABP beta 1L, this subunit, this specific one, it is very important because this is going to do the TERT transcription. Okay, whenever this subunit binds to the DNA, that is going to do the TERT transcription. So alpha subunit binds to the DNA to which this beta subunit is bound, and that is going to do the TERT transcription. And this is how the telomere the telomerase enzyme gets re-expressed into the cell. Or if the promoter mutation doesn't happen, then there are certain epigenetic mechanisms as well. We have already learned this in epigenetics. So the epigenetic regulation of the TERT gene. Let's say there are some histone modifications at the TERT promoter region. The activating modifications like H3K4 methylation, the trimethylation mark, or H3K27 acetylation, those are activating marks. So if those histone methylation marks are set on the TERT promoter, that will lead to the overexpression of TERT and then resulting into the maintenance of telomeres. So this is how the different types of mutations, the genetic or epigenetic mutations, they are going to result into the TERT activation. Let me show you a schematic. So this has taken from this volume. Okay, so this schematic shows us what happens in normal cells and what happens in the mutated cells. So in the normal cells, this is the GABP protein, which has alpha subunit, which binds to the DNA, and the beta subunits, which has different parts, beta 1S, beta 1L, and beta 2. So this is a beta subunit, which is a transactivation subunit of GA binding protein. So this GNA binding protein that can bind to the promoter of the mutated TERT gene. So this promoter is mutated. So this has that C1, what was that? C250, yeah, C250 threonine mutation and C228 threonine mutation. Okay, cysteine to threonine. Those mutation, if they are seen in the promoter region, then the this DNA binding subunit will bind to that mutated region, and to which the beta 1L subunit will be bound. And now this is going to do the TERT activation, which will lead to the uncontrollable cell division. So this is how the cancer cells, they are able to maintain the telomeres. So alongside this TERT activation, so either the telomerase, so what can happen? Either the telomerase enzyme, the gene for telomerase enzyme, that can be reactivated, means the TERT reactivation could be done, or the TERT overexpression could be done by the epigenetic regulations. Or the second mechanism is ALT pathway. These are the alternative lengthening of telomeres. So this is known as alternative lengthening of telomeres. So this ALT pathway, this is a telomerase independent mechanism. So a few cancer cells, they maintain the telomeres through this pathway. So this is a homologous recombination based mechanism, but we are not going to discuss this in detail in this lecture. But this is also an mechanism which facilitates the recombination based telomere elongation. So This facilitates recombination based telomere elongation. This one, this one leads to the telomere sister chromatid exchange and all. We'll learn about this in some other video. So this is how the telomere maintenance is done, which helps in the cancer cell to proliferate uncontrollably. So what can we do what can we do so that the telomere maintenance mechanism can be stopped in the cancer cells? So we can either use telomerase inhibitors. So let's list them down. So we can either use telomerase inhibitors. Okay. Okay, so there are certain inhibitors like BIB R1532. So this is a small molecule which inhibits the TERT, the catalytic subunit. Or then we can use the GA binding protein, GABP, beta 1L sub inhibitors. We can inhibit this directly so that it won't activate the TERT. So targeting this targeting the GA binding protein, we can selectively suppress the TERT expression in tumor cells. And then the third mechanism is by disrupting telomeres. So the TRF1 and TRF2 inhibitors, they will destabilize the sheltering, and this will lead to telomere uncapping. So there are certain inhibitors like tankyarese which impair the telomeric DNA repair. So we can do the telomere disruptors. We can use the telomere disruptors like tankyarese. Okay, so what does it do? This impair telomeric DNA repair. So now, as the telomere DNA will not be repaired, so telomere length will keep shortening, and that will eventually lead to apoptosis or cell cycle arrest. Then we can also use immunotherapies against telomerase, some peptide vaccine that can elicit the anti-tumor immune responses. So we can use there's some called GV1001. This is a TERT peptide. Okay, so this is a TERT peptide vaccine which will elicit the anti-tumor immune responses. Then we also can use the CAR T cell therapy. So immunotherapies are also helpful to prevent the telomere maintenance in the cancer cells. So these are the four mechanisms that we learned for the these are the therapeutic targeting for the telomerase. Okay, so with this we conclude this video. In next lecture we will learn about other hallmark of cancer. Till then, see you. Bye bye.